Good evening, everyone. Am I audible to you? Please confirm in the chat controls. A very warm welcome to all of you joining us today for this exciting and highly informative session. AI ML Toolkit 15 Powerful Tools Biologists Need for Career Success. Yes, who do we have here? Yes, Devi Prasanna. Hello, good evening. Yes, Pavitra. Hi. So, I'm thrilled to see such a diverse and enthusiastic group of students, researchers, and professionals from across the life science community here today. Yes, very good evening, Unitech. Disha, hello, very good evening. So, whether you are curious about artificial intelligence or already dabbling in machine learning, then today's webinar is tailored just for you. As we all know, biology is no longer just about pipettes and petri dishes. In 2025 and beyond, the most groundbreaking discoveries in life sciences, as you would already be knowing, are being driven by algorithms, big data, and machine learning models. From decoding the human genome to predicting disease outbreaks and personalizing the cancer treatments, artificial intelligence and machine learning are now very important essential tools in every biologist toolkit. The best part, you don't have to be a coder and to start using them. You just need the right guidance and tools to get started. That's exactly what this webinar is all about. Biotechnica, your very own channel, is here with an exciting 1 hour session where you would be knowing a lot of things. We are honored to be joined today by three amazing experts who are not only pioneers in their fields but also passionate about helping young scientists build future ready skill sets. First up, we have Dr. Nilofer K. our very own in house expert who holds a PhD in bioinformatics from MIT Edity University. She specializes in big data analysis and computational approaches for canceromics. Her expertise spans Python and R programming, multi-omics data analysis, biomarker discovery, and AI-driven drug design. Dr. Sheikh has published 5 research articles in Scopus Index journals and holds an Indian patent in the field. Today, ma'am will walk us through the foundational AI/ML tools that are reshaping biological research. Our second speaker for the day is Dr. Ishan Pandey, an MSc and PhD in medical microbiology from Motilal Nehru Medical College, Prayagraj, with a specialization in clinical trials and gastric cancer research. He is currently working as research scientist too in a DHR funded multidisciplinary research unit at MNN Medical College and is an adjunct faculty at Savita Medical College, Chennai. Dr. Pandey has 20 plus publications with a cumulative impact factor above 50, including papers with impact factors greater than 10. He is also a reviewer for leading journals like BMC Genomics, Scientific Reports, and IJS. Today, Sir will share AI applications in biomedical domains with live tool demonstrations. And finally, we have Mr. Mallampalli Kapardi, a senior machine learning engineer at Pi Labs and a PhD researcher in computational neuroscience from IIT Hyderabad. His work focuses on neuromechanics, biosignal processing, and AI for healthcare. He previously led the data science team at Breath AI and has presented his research internationally. He's a recipient of the IEEE EMBC Next Gen Scholar Award and was honored at Max Planck Summer School. Mr. Kapade will help you understand how to integrate AI/ML tools into your portfolio and stay career relevant in a fast evolving field. And trust me, you will want to stay till the very end because we will be hosting an open Q&A session at the end where you get to ask your questions directly to our speakers, whether it's about AI tools, career paths, or how to get started with machine learning in biology. This is your chance to gain personal insights from top experts in the field. So come prepared with your questions and don't miss it. Without much further ado, let me now hand over the controls to our first speaker for the day, Dr. Nilofer Sheikh. Over to you, ma'am.
So, a very thank you, Urmila ma'am, and Biotechnica team. Today, I hope that you all of you aware, as Urmila ma'am have given a very broad introduction regarding the importance of ANML and how exactly you guys can bridge the particular wet lab experiments with respect to the very hot topic area and upcoming field, which is really important to integrate with respect to the biological application. And that is nothing but AI and machine learning, followed with the deep learning applications. So today I'm going to highlight some of the most important overview regarding the same. So I'm going to share the screen and then we are going to quickly move towards a quick presentation, and where I'm going to walk through, guys, all of you, regarding how exactly we can think about how to utilize the artificial intelligence with respect to the different areas such as the wet lab way, and how we can bridge the gap. Okay, so as you all can see that, I hope my screen is visible to all of you over there. Yes, guys, all of you are excited. Yeah, because I think we all know that Biotechnica has previously also arranged many kind of webinars where we have discussed about what is mean by artificial intelligence, machine learning, followed with the deep learning application. Today we are going to move one step ahead, where we are going to see that how exactly we can bridge the gap between the wet lab and move ahead to perform the in silico analysis for the same. Yeah, so all of you guys are ready? Yeah, you guys are eager to know how exactly in 1 hour of session we are going to discuss a lot of important information regarding the upcoming areas and how exactly we can utilize with respect to the computational approach. Yes, all of you are ready, guys? So I hope so. Okay, so we are going to move ahead, but before that, let me know, Urmila ma'am, like the screen is visible to all of you guys over there, just for the
Uh,
reference.
yes, ma'am. The screen is visible. Please continue, ma'am.
Thank you. So as you all can see that the first slide is indicating to all of you like how exactly we can move ahead to transform the ANML tool in biology. So it's nothing but where we are trying to explore artificial intelligence and machine learning majorly. And definitely a deep learning will come into the picture, but it will depends on how exactly you guys handle and get an hands-on experience with respect to this kind of techniques such as machine learning, right? Because whenever we are talking about big data, right, wet lab experiments and the techniques, ultimately the data will come in the form of raw format, right? All of you agree with me, right? So if we are handling a data in the form of raw format, so definitely we need certain platforms where we can process our data smoothly, and today it's possible. So definitely one step ahead, we are taking help of bioinformatics tools and even the pipeline to process our data smoothly. But nowadays, hand in hand, we have machine learning and artificial intelligence tools and technique to process our data smoothly. So in this context, we are going to see how exactly we can use ANML, where it is getting a one of the revolutionizing field with the biological research majorly, and it is bridging the gap, right? Where we can think about the traditional approach and move ahead with respect to a groundbreaking research and even the discovery with respect to the biological field. Moving ahead, the introduction over here, definitely I'm not going to move ahead in very much in depth with respect to what exactly mean by artificial intelligence, what is mean by machine learning, followed with the deep learning, right? Because many webinars we have conducted where we have given you a broad overview, but here the aim of today's webinar is to show you guys like how you can think one step ahead, right? And majorly if you guys are working in the core area is nothing but the specific disease topic, and you are very much interested to go ahead and perform the wet lab experiments, right? So in that context, in this particular field of ANML, you guys can start your research with computational approach and end up with validations such as wet lab, or else you can even do your wet lab findings and do your predictions with respect to the ANML approach. I hope you guys, you're getting my point, right? So these are the two way of approach where you can take this particular platform and then you can do lots of possible analysis and groundbreaking applications in your research findings. So as you can see that how to explore the ANML. So in this context, we need a data, because ultimately nowadays, whatever the wet lab experimental techniques we are handling, it is all dealing with respect to the big data processing, right? For example, if you're handling the NGS data, so it will come up with respect to the enumeration of very high sequencing data. Maybe if you're using a genome sequence of a human, or else if you're interested with any other species of your interest. Anyhow, the data which you're going to handle is based on the sequence. So in that context, we need certain platform where we can handle a big data and in the way of smooth analysis. It is not only dealing with the analysis point of view, but also to find out the particular hidden patterns and play around the world of biological research, right? So remember, guys, this kind of fantastic things we can achieve by integrating the ANML with respect to the computational approach, and the data which is coming is nothing but from the experimental techniques. So as I given, the one of the example is the next generation sequencing data. Apart from this, you can even take ahead the multiomics data sets in the form of proteomics, where we have the quantitative and the qualitative data to get screened and do the predictions of the markers identifications, so that in future we can even design the antigen antibody designs with respect to the drug target identifications, or else we can identify the different vaccines for the new or else the upcoming outbreak kind of disease. Yes, we can predict those things by integration of certain techniques, and that is exactly what the aim of today's webinar is, to show you guys like how exactly we can bridge the gap. We can look towards the particular wet lab analysis, towards the validations, and move towards a more prescribed predictions by taking help of ANML for the same. So complex data, what we are handling is coming and enumerated from with respect to the experimental techniques in the tool. And apart from this, we are looking towards enhancing the drug discovery process. Because as you can see, guys, okay, so many type of diseases are there, right? And even the drugs are existing into the market. But still, we are continuously working in the research area to find a more potent drug target. So why these things are happening, right? So it is totally depend on the drug resistance mechanism, or else the genetic or the phenotypic condition of individual patients. So this is exactly what the core area where it is depend on. We have a bulk of data sets, right? We have enough of resources in our hand, but every researchers used to think about how exactly we can move one step ahead and deal with respect to the novel finding. And this is exactly, guys, where ANML will help you to think about that scenario. It's nothing but we can work on the drug discovery process, where we can come up with respect to the bioactivity predictions, designing the different models such as the toxicity models, or else you're looking towards the bioactivity models apart from the Lipinski rules, or else you're looking towards the drug properties, QSAR models, prognostic models, or else the predictive models for the same. Yes, we can achieve those kind of findings by integration of ANML. Whereas if you can see over here is nothing but predicting the protein structures. I hope you all of you are definitely aware, like one of the very tool which is available, okay, and we guys are using, is nothing but the AlphaFold tool, right? So it is totally an artificial based prediction tool. And even you can see that previously we have AlphaFold, then we have AlphaFold 2, and now we are using AlphaFold 3 also. Why? Okay, because as per the requirement of the data, and as per the come up with respect to your novel findings, even the databases and the data is getting updated based on the requirement of the data sets. And that is exactly why the whatever the experiment analysis we used to do, we can even predict the protein structures for the same. And these structures are totally depend based on the artificial intelligence algorithms. Moving ahead, how exactly we can transform the role of ANML in biology. So in that condition, as you all can see that the one of the most important challenge in every research is that handling a data, okay, and even managing that specific data accordingly, as I mentioned earlier. So if you're handling any kind of experimental data, so anyhow you're working with respect to the raw data. So the first important process is to handle a data set in the form of the volume. So volume is nothing but the type of data or is the size of data sets which you are handling. So it will get specified with respect to the data sets which you are interested to process accordingly. So in ANML, one of the most important pipeline to process your data is about data volume management. So ANML process will handle a large data, and it will help us to uncover the insights beyond the manual analysis. Yes, we can do this kind of stuff with ANML. So for example, if you have a millions of data points, right, if you have a sequence of for example, with respect to your individual patients, right, with respect to their gene and expressions value, or else you have a data of millions of patients with respect to their sequences, right? So it is very challenging even for the researcher to manually curate all this stuff. So we need to take help of certain smart technology and even the techniques to process our data and uncover the hidden patterns in the biological world. And remember guys that it is achievable, right? If you just go ahead and visit certain research papers which is get published in recent year, so you will definitely find that certain kind of research has been done, right? And the papers and the research is getting published. So that will be one of the strong template for you guys and even the upcoming researchers to think about how exactly we can achieve this kind of goals with respect to the integration of ANML in a biological research. Moving towards the next important pipeline, whenever we are dealing with ANML, is to enhance the predictive accuracy. So, how exactly we can enhance the predictive accuracy? The simple answer is that based on the literature survey, right? For example, as I mentioned, AlphaFold is one of the protein structure prediction model available, and many of the researchers are using the same. So why exactly the AlphaFold was there previously, and then we have AlphaFold 2, and followed with that we are using right now AlphaFold 3, because it is looking towards what exactly the new features or as the new predictions we can enhance so that it can look towards more personalized finding. And that is what you can see over guys over here, is that ANML is not about like taking the data, processing and doing the predictive analysis, but you guys can even enhance its accuracy. How exactly we can enhance is nothing but studying more data, integrating with respect to the relevant features, and training with respect to the multiple models with the accurate findings. And in that context, where is exactly the role, where exactly the application of certain enhancing accuracy is nothing but where we can predict the protein fold, and even we can study the drug efficacy. So that is exactly one of the quick example, just to note. Moving towards the next is nothing but increase the resource efficacy. So increase the resource, why we need certain resources. Resources are ultimately where the researchers can follow the specific pipeline, okay, in the form of the data set, or else in the form of the script or the command, right, or else the algorithms to process your biological data in the form of data analysis, and even to come up with respect to the predictions for the same. The next thing what we can do is nothing but facilitations of the innovative discovery. So innovative discoveries are nothing but the novel discovery, because I just saw about like many students who are very eager to do research in ANML field, right, or else in any research area. They're looking towards the first question like, ma'am, how we can do the novel findings, right? So this is the common questions we used to get to know from every students, even the researchers for the same. Or else sometime you will get to know like, this is the one of the thing we used to find like, or else we used to ask the question to ourselves, right? How I can achieve a novel findings based on the known research or the data. So the novel findings or the innovative findings are nothing but this pipeline which we have discussed right now. It's nothing but looking towards the data sets, okay, checking the which type of data you are handling, okay, and then you are looking towards the increasing the specific resources to smoothly process your data, and even in that context, how we can enhance the predictive accuracy so that if we can look towards a more personalized findings for the same. Because in future you can see that whatever the era is depend is to deal with respect to the personalized medicine, right? So nowadays we are finding the biomarkers, we are saying that it's one of the potential drug targets, right? And in the conclusion of every research paper you can see guys, like based on multiomics analysis, it has mentioned that this is one of the precision therapies in future can be applied. So ultimately, in this era where we are handling all the big data, we are processing, curating, and we are storing the data in the form of databases. So that after once the databases are created in the form of proper data management, so the researchers can process the data in a meaningful manner because already the machine learning analysis has been done. Only the thing is to look towards how we can increase the efficacy with respect to their resources, and even with respect to the predictive modeling is nothing but enhancing the accuracy. So in that context, there is high chances that in future we can even get a drugs in the form of precision or else personalized therapy. Yeah, that is possible because nowadays the data is coming with respect to your real time screening. So we used to get the data from smart sensors, right? Biosensors, okay, real time patient data sets. So this data we are able to capture, and to process the data and to lead with respect to the analysis is nothing but where the ANML is taking step ahead to process your data smoothly and do the predictive analysis, right? So I hope that all of you are getting the insights what exactly I'm talking about. So basically I'm just trying to give you a small hints over here because itself is a very complex process, okay, and that's why today we have a webinar to give you a quick insights and even the hints, right, how you can grab those kind of opportunity if you just be very specific you with your research process, right? Moving towards the next slide is nothing but how exactly we can integrate the wet lab and the computational skill, because that is the main aim of today's webinar, right? So if you are a biological researcher, core biological researcher, okay, where you have not handled any coding experience, okay, you don't know how to process your data with respect to even with bioinformatics approach, no worry guys, okay. So we are here to guide you with respect to the same process. We are here to tell you that how exactly you can bridge that specific gap and you can integrate your wet lab data with respect to the computational tools and techniques, right, followed with definitely A and ML. So here you can see that collaborations with between the biologists and data scientists. So basically, if you just see guys, is nothing but to understand how the wet lab data can be handled with the computational processing. It's nothing but you have to discuss, right? You have to have a good collaborations where you can enhance your skills, and even you can think about how you can upgrade your skills with respect to the processing of biological data. So in that context, we need interdisciplinary works where it can be enhanced with the data interpretations. See, guys, if you have a biological data based on different experimental technique, but if you don't know how to interpret your data, that will be having a huge challenge. Because if you have something which is very novel, but if you're able to come up with respect to your novel findings or as the predictive analysis, so that is exactly where you have to make a good collaborations where you can enhance your skills accordingly. Next thing is nothing but experimental design optimization. So as I mentioned, like if you're handling any wet lab data and you're referring the same protocol, right? But if in the lab or is the in the research we have a different upgraded technologies are coming into the picture, so why can't we use certain technology for the same, right? So as I mentioned that next generation techniques we are using, right, followed with that previously we were handling Sanger sequencing, right? So we have upgraded, okay, from Sanger to the next generation. Next, nowadays you can see that we are also having a chance to handle a nanopore sequencing technique, right? So that is also best things we can be achieve. So that is what guys, the wet lab researchers who are attending today's webinar, I want to show you guys like I want to just tell you like one of the important hint over here. Like these are exactly where you can think about upgrading your experimental stuff with respect to the upcoming techniques and the tools, right? And again, one of the upcoming technique what we have is a CRISPR-Cas9 technology, right? So how we can target the specific genes according towards the CRISPR-Cas9 technology, and then do the target oriented predictions. So that is also one of the field where we can think and work accordingly by experimental stuff and optimizing our designs so that the simulations will allow us to do a better planning and the resource managements for the same, right? So you will have the proper timings to process your data. You will have the recent techniques and the tools to come up with more prescribed findings. So that is exactly what we need in the research. Next thing is to enhance the data analysis and capability. It's nothing but if we take help of ANML, okay, this is exactly where we can take help of computational tools. So we can take help of ANML for analyze the data and accelerate our research with respect to the results and analysis for the same, where we can enhance the predictive analysis or else we can look towards more specific modelings for the same. Okay, I hope guys you are getting the insights for the same, right? All of you are agree with me, right? All of you are able to make sense like whatever this flow is going on, right? So this is exactly what you guys have to think as a researcher, right? So definitely if you are a biological student, okay, if you're a non-biological student, no problem. But whenever you're thinking with respect to the data set, so ultimately you have to consider yourself as a researcher. Then only the novel things will come up accordingly, right? So this is how we can take help of ANML to enhance the analysis of biological data. Next is to scaling. Okay, you have to upgrade your skills with ANML, and you have to understand how exactly the pipelines can be generated. Definitely one of the things can be done by guiding you, okay, in the form of like any projects, right? Or else we can guide you with respect to the understanding the pipeline, right? But apart from this, the hands-on analysis and the experiments are really important, okay, where you can upgrade your skills and work with a specific core area. Got it? And then the next thing what you can do is nothing but you can look towards the literature survey. You can perform certain literature survey which will help you to understand what exactly the current research is going on and how this new techniques I can implement in my research for the same. Okay, so I hope that you all of you get the insights in this slides. Next important thing which we are discussing is that how this ANML tools we can use. Okay, so if I'm talking in the last slides about like you can enhance your predictive analysis or else you can increase your accuracy for the same. So in that context, the ANML tools what majorly we are using in the computational approach are nothing but Python, right? So these are the quick programming languages. But I want to tell you guys that whatever the languages has been mentioned on the screen, it's very simple to use. Okay, this languages has been created on the basis of thinking like how a biological researchers can handle this data and then process with respect to the programming skills. So the first language which we are using is Python. Okay, so Python is one of the very simple language where we can do a simple coding with respect to the biological keywords. Remember, guys. Okay, so if, for example, if I want to do a sequence analysis, so we have certain sequence analysis scripts available with us. Okay, there is nothing called as a coding, but it is all about with respect to the codes are written with respect to the biological stuffs. Okay, and then we have a biopython. Okay, see, Python is a programming language which help you to do your data analysis and data visualization, and some of the things you can even do the predictive analysis for the same. But biopython is a certain package available where you can properly analyze your biological data accordingly. Okay, then this all the things you can do guys using the different environments. Okay, the one of the environment is nothing but Jupyter notebook environment. So what is mean by now environment? Environment is nothing but where you can write your commands and scripts. Okay, if you are all remember guys, right? Previously if anybody have used any kind of coding languages such as Java, C++, okay, so you all of you guys have definitely seen the things called as the CMD command, like a black screen or the white screen, right? And there we used to write the commands and the stuff. But we don't know like what exactly the commands are running for what particular purpose, right? We were totally like unaware about that stuff. But here, Python programming and followed with that R, you can see over here. So these two languages has helped us to process our data smoothly because majorly the commands and the codes which are written in the form of biological understanding. Okay, so if you're using R programming, then Galaxy is one of the software will help you to understand how the multiomics data, such as genomics, okay, majorly RNA sequence analysis, right, FASTA-Q files in the form of next generation sequencing, can be analyzed, processed, and even do the visualizations for the same, with respect to a step-by-step guidance, and even the protocols has been shared. So you guys can definitely explore these tools for the same. Next, we have a deep variant. So deep variant is a specific packages are available where you guys or the researchers can specially work on the genomics data analysis. Okay. Followed with that, we have a bioconductor. So as we have in Python called as a BioPython, which deals specifically with biological data analysis using Python packages. So in the similar way, we have bioconductor with R. So in bioconductor, we can do all the biological data analysis, starting with respect to the sequence data, omics data, multiomics data, genomics data, right? Or as the network analysis, drug discovery, and all the stuffs we can carry out with respect to the bioconductor package. The only thing what we have to do is to install the required resources, and then we have to proceed with the proper environment by installation of the required package. And definitely one of the important data is nothing but the type of input data we need to process for the same. Okay, so I hope that this slides help you guys to understand how exactly we can bridge the gap between the wet lab and computational, and integration of ANML for the same. Okay, moving towards the next slide quickly, is nothing but the fundamentals of R and Python in the biological world. So why we are using these two languages only? Okay, because these languages are user friendly, because the syntax which are written is nothing but the command which I've explained in my previous slide, right? So the command and the scripts are written in the very simple way where a non-programmer or the biological researchers can quickly understand, and they can adapt that language accordingly. That's why it is called as a user-friendly syntax are there. Okay, where you can learn and make them accessible for the biological coding experience. Is that true, guys? Okay, all of you are clear. Next, the use of these two programming languages are nothing but extensive libraries. Okay, extensive libraries are nothing but as per the requirement of the biological researcher. For example, I'm interested in the genomics data analysis, so we have the specialized packages are available to process your genomics data. Okay, so that is really good, right? So if I'm interested with respect to the drug discovery, so yes, we have specific packages designed to process your drug data. I can draw the 2D structures of drug, and even I can convert the 2D structures into the 3D formats. So virtually it is very a nice experience. Okay, and you guys will definitely learn the things by having the overall biological environment on your screen. Okay, so in that context, extensive libraries has been generated, such as biopython and bioconductor, okay, and so on. So for data visualizations, we have ggplot for in R, matplotlib, or the plotly in Python, and so on, right? Then third important thing to use certain languages is that it has a strong community support. We can have a large community environment where we will get to know how to process the data and then do the problem solving environment. It is one of the versatile application, because we can even integrate this programming language with respect to the ANML use, where we can do the machine learning analysis, and even we can perform the predictive analysis for the same. Okay, so here you can guys can see that this is exactly where the Jupyter environment is there. Okay, with the Python, where we can perform the interactive environment. Okay, why it is interactive? Because whatever the commands we are writing over here, right, it is a line by line command writing and doing the executions for the same. So we used to know, and we should be aware like what we are writing in the command and what kind of outputs we can expect from here. Okay, the next thing is that we can do the integration of the codes with the documentations. For example, if the overall pipeline is ready with the genomics data analysis, so you guys can definitely make a documentations of your codes. Okay, so nothing will be get wasted. All the process will be properly documented in the form of code, followed with the data visualizations and even the analysis for the same, so that you can share your entire script with respect to the further process and even for the further predictive analysis for the same. Okay, so we can even reproduce the script. For example, we have designed a genomics pipeline and we want to do certain kind of biomarker predictions in that. So yes, using the same script, we can reproduce the command by with respect to the integration of biomarker predictions. So that is again a fantastic things we can get from programming language such as Python over here, right? And the versatile in the outputs are nothing but where we can save the entire script in a different outputs such as document, we can save in the PDF, HTML, or else specific data visualizations format in the PNG, okay, TIFF file format for directly providing for publication quality work, or else we can save the script in the PY file format. Okay, fantastic, right? So these are the things we can achieve. Talking about R, so R is again one of the comprehensive bioinformatics tools can be processed with respect to the R, followed with definitely BioPython, because BioPython and is a specializedly designed to process your biological data. Okay, so R is also included with that where we can take help of bioconductor, but here in BioPython we are using to process the complex biological data. In that way, that data is get pulled from the bioinformatics resources such as NCBI. We can work with respect to many algorithms, okay, to implement and do the sequencing and structure predictions. And it is also one of the community distributions. It's nothing but where we can contribute our findings with respect to the other researchers who can use the data or the script and do the further step of data analysis. Okay. Whereas here, Galaxy, as we have discussed, right? Because Galaxy is one of the software which is available, okay, and it's a web-based application, and followed with that, you can even download for the same. Both the things they have provided. So Galaxy is a software where it's again is a user-friendly. It will handle a complex work process and it can be accessible. And if you're a non-biologist, okay, computational biologist is nothing but who don't know about like how to handle even a bioinformatics resources, they can even understand how the process can be run for the omics data analysis. It is one of the integration tool because here you can perform the data enabling with the complex data and then processing ahead with the programming skills for the same. You can even reproduce the workflow, and then you can even look to enhance the workflow so that the workflow can be even reproduced in the research quality work. And it do have a community support. So community support is nothing but a different tutorials are given to process your multi-omics data such as genomics, okay, NGS data, proteomics, and so on. And it supports that new users to give a step-by-step guidelines and adapt how to use that specific um, environment for their research point of view. Okay, so I hope guys you are getting the insights for the same. Next is deep variant. So deep variant is specifically used to process your genomics data. So here also where we can highly look towards the accuracy of variant identification. So variants are nothing but the type of mutations, right? So in that we are finding the different type of mutations accordingly, but using deep variant. Why the name is given deep? Because here the deep learning algorithms are used to identify the different kind of variants in your genomics data processing. So deep learning variant or the deep variant will help you with the integration of deep learning and find the prescribed variants identifications with respect to the data sets which you are handling. Got it? It will even efficient your data handling to process the large omics data sets because as I mentioned, genomics data itself deal with respect to the big data, large sequencing data of the reference sequences. It's again a user-friendly interface where the upcoming researchers and the new users can quickly adapt and they can perform their research accordingly. It's an open source community driven where you can upgrade your skills and you can perform a novel innovations by taking help of this particular process. I hope guys you are getting the insights for the same. Next is bioconductor where R has been used over here because as I mentioned in my previous slide, like R is also one of the programming language which is widely being used by the biological researcher. So you guys can see that R for bioconductor is specifically designed to process, analyze, and work your bioinformatics data. So in that we have an extensive packages repository such as bioconductor, which can help you to process the RNA sequence data in the form of single cell RNA sequencing, bulk cell RNA sequencing, okay, genomics data with respect to the omics data analysis, right? Or else you can even work with respect to the microarray expression analysis, right? And so on. So certain kind of analysis you can perform because the packages have been designed. So now the question will be like, what is packages? Packages are nothing but the pipeline, okay, starting with the raw data towards the research findings. All the steps are given in a proper flow, okay. It have active community support. That is one of the great application in every programming. You will get a community support to understand and tackle any kind of errors if you are facing, how you can resolve the same. You can even integrate with respect to the R to perform the statistical analysis, because in research statistical analysis is really important, and we can even reproduce according to the objectives what we have as per the research finding, okay. So overall the AI application in the biological research is that AI can reduce the drug discovery time, okay. So pharmaceutical companies has a huge requirements for certain candidates or the researchers who have the skills to understand how exactly the traditional approach of drug discovery can be overcome with respect to the ANML. So I think guys who are attending today's webinar, you guys have a very great opportunity to learn those things and implement and be ready for the upcoming future opportunities for the same. So you guys can call yourself as a ANML researcher, right? You can be yourself as a data scientist, okay? So certain kind of opportunities are awaiting. So it is really good to understand and implement this kind of flows or as a discussion what we are doing in today's webinar, right? We can even do the improvement of the diagnostic accuracy, because that is really important, right? So the accuracy is really important even for the diagnostic and even do the further predictive analysis. So AlphaFold is one of the real-time tool which we all know and we guys are using for the protein structure predictions, and we are understanding the disease mechanism for the same. So ANML tool is also helping in the medical imaging where we can diagnose the different diseases accordingly. So the challenges, definitely every technique will have the challenges, but we have to think about how exactly we can overcome the challenges. And that guys you can understand and learn if you just understand the overall pipelines and work with respect to the specific projects for the same. So data quality, data availability is one of the highest challenges, but definitely the challenges can be get overcome if you know the standard pipeline. Complexity of the biological data. So from raw data, how you can do the pre-processing, you can do the data curation, look towards the feature engineering to find the specific features and the or as the data which can be trained ahead and do the predictive modeling. Skills gaps are there because definitely today we are discussing the same thing, right? So how you can upgrade your skills with the computational tools and the technique so that you can focus specifically with respect to the biological finding. Ethical concerns will be there because if you're predicting any AI based approach, so we need the data, so the data will be in the privacy, algorithm bias will be there, so that's why validations, cross validations, okay, data curation and the control access of the data is one of the challenges over here. So definitely the challenges will get overcome if you just go and refer the standard protocols and the pipelines for the same. Okay, it sounds good, right? All of you are getting the point, right? All of you agree with me? Yes, I hope so. So last slides are remaining only two. Okay, we'll just quickly wind up and we can move ahead for the same. So as you can see, guys, okay, you can compare that only in the last 3 to 5 years, right? You can see that how ANML is getting practically, like it is getting on and it is one of the upcoming field with every domain. But today we are discussing that how we can integrate with the biological stuff. So ANML has integrations with a more predictive finding, right? It has a very enhanced the collaborations of even every researcher and even the startups and the industries, right? So there also different companies are adapting ANML to do more prescribed and predictive findings. So real time data is even getting pulled out and screened with the biological finding. And ultimately, the future application is that advancement of personalized medicine, as I mentioned in my previous discussion. So why we are doing all this stuff? Definitely, real time we are finding the biomarkers. We are looking towards the novel drug targets, right? But in future, we can look towards designing the personalized medicine. We can even look towards the any kind of outbreak kind of disease we can predict for the same. So that is exactly what the core application of ANML. I hope that you guys are understanding the concept, right? And how you can upgrade your skills. So definitely Biotechnica is one of the platform for you guys, right? So you can explore certain free or the paid courses. One of the platform what we all know is Biotechnica. So it will help you to enhance your skills even you are beginner. You can attend the workshops and even the seminars, okay, to understand the overall core pipelines. You can get a proper mentorship opportunity so that you will get a step by step guidance that how you can achieve certain goals and be ready for the future career opportunities, okay. And all the things are depend on your practice and understanding the experimental data and even the protocols for the same, right? So the empowering your particular research with ANML is nothing but where you guys have to integrate the ANML with the biological research and think about how the groundbreaking research can be enhanced with the collaborations of ANML. So I just really all the best to all of you guys who are awaiting and really eager to know practically how we can achieve that stuff. I think the next speakers will definitely guide you for the same. So that is from my end. So I hope that you guys understand the quick overview regarding the how exactly the ANML can bridge the gap with respect to the wet lab and upgrade your skills with the computational power. So all the best to all of you guys, and I hope that the concepts are clear from all of you. Right? Thank you once again. I just request the next speaker to please proceed and please explore accordingly with respect to some practical insights for the same, so that our audience can understand how we can achieve certain goals
Thank you so much,
accordingly.
Dr.
Thank you.
Nilofer. Thank you so much, Dr. Nilofer. Can you hear me?
Yes, sir, you are audible. Please continue.
Madam, ma'am, can you hear me?
Yes, sir, you are audible. Please continue, sir.
Yeah. Yeah. Is my screen visible? My slides are being visible to you all?
Yes, sir, the slides are
Hello.
visible. Kindly make it full screen.
Okay. Yeah. Is it visible in full screen mode?
All good, sir. Please continue.
Okay. So my introduction has already been given to all of the participants and to all of the organizers. So let me continue my presentation as I will be, rather than the experts who are giving their talk on how to develop the AI ML tools and what are the advancement in AI tools. Being a medical microbiologist or a biomedical scientist, not having more of the computational background, I will just highlight the implications or the uses in biomedical research, how this AI and ML is transforming. So I will be more of the user friendly background. So I will be sharing my experiences how using these tools have enhanced it. Okay. So what about the introduction to biomedical research, introduction to biomedical AI and ML? That AI ML brings innovation in diagnostics, genomics, and personalized medicine. Means what is about genomics is about the genes or the sequences or about the genetic information, how the disease, because nowadays the diagnosing disease has not become a universal protocol. It has become very much personalized. You have to be it varies based on person to person about how their genetic sequences are there, how their genes are responding to the environmental factors, to the other risk factors. So this AI and ML, or the artificial intelligence and the machine learning, is giving a personalized treatment or the personalized diagnostic, how this treatment or the diagnostic can vary from person to person. It assists in disease prediction. Now there are three components. One is the disease prediction or the disease, knowing about the disease or the descriptive science. Then there is the drug discovery, means the discovery of the treatment. And then there is a what how this AI in ML uses this disease prediction and drug discovery is about the pattern recognition, because when we train the AI and ML software, it gets a pattern recorded in its system, and according to the patterns it creates a algorithmic system to recognize the disease and create the drug discovery. Now it integrates, now this AI and ML integrates all the omics, that is genomics, proteomics, lipidomics or metabolomics. All these omics are being integrated with the use of advanced imaging diagnostic. Now there are lots of pictures or the image processing tools where we can process the images. Maybe you have biological images and the data for biological images is in lakhs, is in thousands and lakhs. So analyzing these images, whether it is a CT scan or an X-ray or a CTHR or any kind of pathological diagnostics, histopathological slides. So this clinical data, there is a lot of clinical data which is being visualized in the form of pictures or photographs. Now I'll be taking two most important tools. That is, one is the KNIME and then the other is a cell profiler. Now what about KNIME is this is called Constance Network Information Miner. So this is a workflow, visual workflow based data analytics tools. It uses it tells about the gene expression, the biomarker detection, integrative data analysis. So it gives you a workflow or a flow chart which gives you the analysis about the gene expression, how the gene has expressed, it is overexpressed or underexpressed, what are the biomarkers, means on the basis of expression of these genes or on the basis of expression of some proteins. Maybe the gene has amplificated or the protein has overexpressed. So based on this analysis, it gives a workflow, and it on the basis of integrative data analysis. Then there is a cell profiler. As the name suggests, that it is about the profiling of cell. This is the image processing tool. Now it quantifies the biological images. Now what we see in an image, we see a larger image. Then we zoom it. Then we go to the more deeper analysis, and we see that whether that image is producing something notable as a change in the sequence, so that we can profile for phenotypic screening rather than the genotypic screening. So based on the phenotypic screening, whether the if we stain or immunostain a dye, that is we are doing an immunohistochemistry procedure. So the immunohistochemistry staining will give a deeper insight, and the surface markers will stain more on the slide. So that can be visualized in the image based cell profiling and phenotypic screening. Then there are three more architectures which are supporting this AI and ML. Broadly, they are ANN, that is the artificial neural network. Then there is CNN, which is convoluted neural network, and then there is RNN, which is the recurrent neural network. Now, ANN is the basic model for disease prediction. Then the CNN is applied in medical image analysis, like the X-ray, MRI, histopathology, etc. I told you in that. So that is basically on the diagnostic part. And then there is the RNN, which is suited for time series data, example ECG, genomic sequences. Then there is voice recognition, pattern recognition. This is for this. Then the use of CNN. Now there are case studies and demonstration ideas, which is now how we can use it. Now the CNN is to use to classify histology sites. Now histology sites, based on the WHO classification or based on the Lawrence classification, it can be classified into poorly differentiated carcinoma, or if we are talking about cancers, because my main work is in gastric cancer. So if we are talking about poorly differentiated carcinoma or well differentiated carcinoma, there are different kinds of cells are present. So how this histologically or based on the tissue basis, how we can classify those slides, it can be done. Now, AlphaFold for structure prediction in drug design has already been told by Dr. Nilofer previously, and it was a detailed talk. Then KNIME, I told you that KNIME is about the workflow demo. The gene expression classification pipelines have been used in KNIME. Then cell profiler is about detecting morphological changes in the treated cell, whether you stain a cell, whether you treat a cell, you give and any kind of drug, and how the cell has been reacting and how the morphological changes have been that. Everyone might have heard about this ImageJ or a Fiji. That is a basic tool for image analysis, and there are many plugins also there, that IHC profiler, cell profiler. You all can add these kind of plugins in ImageJ and can use very well. So apart, I have used my one of my my papers where I have used all the artificial intelligence based technologies and the molecular and compared it with the molecular techniques used in gastric cancer detection, the purpose of the AI and the references of the last previous 10 years or the 20 years. So as you can see that if you are using fluorescence in situ hybridization as a molecular wet lab tool, there is a artificial intelligence based technology in the second row, that is jellyfish. What it does, it enhances the accuracy of fish results. So what happens by using the AI and ML tool is the accuracy, the time, the the time frame and the accuracy of the results have been enhanced. Now then there is, I was talking about immunohistochemistry, that is the IHC. There is immunomembrane, which gives the accuracy of immunohistochemistry results. Then slide imaging, whole slide imaging or the PG is about microscopy and histopathology. Then again immunohistochemistry, like the cell profiler, there is IHC profiler. It enhances the accuracy of IHC expression of proteins. Then IHC fish RT-PCR Western blot. These images have been used by CNN or the convolutional neural network classification and detection of HER2 plus tumors, HER2 tumor or non-tumor, necrosis detection. These all can be done by CNN architectures. Now, slide imaging, whole slide imaging, image J, plug-in slide J, microscopy, and histopathology. Then slide imaging is also being done by QPath. QPath gives you how much tumor content is present inside the tissue. Then there is again fish fluorescence in situ hybridization can be used by Spotland. It also enhances the accuracy of fish results. Then there are 3-4 slide imaging softwares that like the residual neural network, CNN architectures, and CNN with deep generalized multi instance learning. So these all gives the difference between gastric cancer type, intestinal or diffuse type, gastric cancer or non-cancer, classify differentiation degree poorly or well moderated, as I told you, Laurent type, intestinal and diffuse, different type of classifications are there. Then there is a detection of singlet ring cell carcinoma or the component carcinoma. Then these classifications or these intricacies involved in the detection or diagnosis of this can be very much reduced, and the observer can be 97 to 98% helped in giving the diagnosis in much faster time than it is done at an interpersonal level. Then there is fish slide imaging. There are 3 more softwares, and one is ResNet or the ImageNet. Then there is CNN for benign and malignant stomach biopsy specimens. And lastly, the KNIME can also be used for fluorescence in situ hybridization, which will give the detailed results. Then what about the future directions and conclusions about this fusion of AI with the real world clinical decision systems? The most important and the most important thing, that AI will not take the job of any real world clinicians. They will only assist and assist in more accurate manner, more faster manner, so the disease diagnosis or the treatment response can be very fast and can be multiple times manifolded, so that the decision can be fast and the cancers or the diseases can be predicted very early, and the treatment can be started at very early stage, and there can be a better prognosis. Now the interdisciplinary collaboration for translational research is there, like we have to collaborate with all the engineers, all the biomedical scientists, different kind of bioinformatic experts, biomedical engineers are there, computer software engineers are there, electrical engineers. So there is a amalgamation of lot of interdisciplinary collaboration for translational research. Then there should be the focus on explainability, reproducibility, and validation. So there are lots of softwares, open source softwares are being developed by different kind of NIH or the other kind of agencies like the OSDD of CSIR, and these all have to be much more explainable, much more user friendly. They might be reproducible, that is they all every time they have to reproduce the same data. It cannot be variable up to the interpersonal level of different users. And the validation, that it should be validated in the real world scenario. Then the AI final is that AI in artificial intelligence and machine learning to lead biomedical innovation in coming decades. Now this is all about my talk, and thank you all. I hope I'm clear. This is my contact, and I hope I'm clear.
Thank you so much, Dr. Ishan, for the insightful session. We uh, we will be uh, we will be calling you back during the Q&A session for our students for if there are any queries. And up next, we have Mr. Kapardi with our next speaker with the next session. Over to you, sir.
Hello, good evening, everyone. I hope my audio is clear enough. And please give me a minute. I will share my screen so that I hope my screen is as visible.
Yes, sir. The screen is visible and your audio is also clear.
Yeah. So thank you, ma'am. Thank you. So, good evening once again to everybody. So, I would like to thank the earlier speakers for their insightful session and sharing their knowledge and time with us. They have mentioned certain very key aspects, and I hope that will be very useful for everybody who in the upcoming future. The key aspect what they have mentioned was preparing applications and programs that would help you to do research and understand the data, performing analysis on the biological data and so on. I would be talking about these specific tools, the resources that are actually offering us, and probably some of a good platforms that would enlighten us about these tools and the necessary skills that will help us to upgrade ourselves to perform a meaningful research and do a great strides of progress in biology. So, having said that, uh, uh, first and foremost, to start with, we all have a lot of career aspirations to use AI or machine learning in biology to take the lowest hanging fruit and use it in our research so that it would bring out a good real world application and an impact in what research we are doing or the passion that we want to chase on. But to do that, we need requirements of certain skills, certain resources, and also a great amount of teamwork, which involves people from very different backgrounds. So, having said that, one of the most important aspect when you come to machine learning and or any computer aided programs that you want to use for research, maintenance of those programs or applications is very important. You can consider this or correlate it with something like your project report or a library open access system that would actually have a contain their entire repository or the data about the materials or the things that have been done over a period of time. So for the computer aided programs or whatever this is, uh, the most important platform that actually provides this option is for you is GitHub. So what is GitHub and what is its significance? So if you see, this GitHub is being widely used by international and national enterprises, companies, even colleges and students, everybody who is into programming or use computer aided programs. So what this does is it's just like your project report. You know, you say you have done a particular experiment on so and so date, and you want to save it. You want to actually document it. You want to maintain a record of it. So GitHub exactly allows you to do the same. Say you're writing certain programs. It can be in any environment. It can be a notebook. It can be a standalone program. So you can actually save that program in your particular space. You can write a small README for it, saying that what this program is used for, and what is its purpose, and what are the corresponding inputs and outputs for this. And you can also maintain a track of it. Say you have started something you have just started to write up another aspect as well. Then you can record the program version, saying that this is the first version of my program, which will help me to satisfy a singular objective. And then moving forward, it will actually then this is the second version of program, which will help me to do both the activities A and B together. So you can do a version tracking of this. You can make your programs more readable, reusable, and also you can share it across to your friends, your acquaintances, so that they can use it, they can comment about it, they can help you to improvise the program structure. So GitHub is like the DMART, equivalent for maintaining all these records and the repositories for the computer-aided programs and your code project structure. A sample, I just want you to show how this is being maintained. This is a GitHub profile of a very famous computer scientist called Andrej Karpathy, who headed the Tesla's AI team. So if you see, he has a project called Micrograde. So he has uploaded an image and he has given what the engine does, what is the description of it, how to install the necessary components to to use this particular program, how to use this program, what are the relevant sources, and what is the code for it, and then what is the purpose of it, like how it is being done, what which particular program you have to use. Say, do you have a notebook, or if you want to use a particular neural net, what is a library package for it, and a sample result, so that when a person actually comes to see sees this particular page, he will get a basic overview of how to install, how to use a particular program to achieve his objective. So he has an input, he has a description on what the program does, and then he has a sample output or an expected output using that particular data. So let us see how actually to initialize GitHub. It You can use it in both your Windows system and Linux systems. You can have a particular GitHub desktop version. The GitHub desktop version would look something similar to this, where you have your current. You can list out your current projects, what you're doing, and it will help you to actually tell what are the changes that have been done. Say, for example, something in red, it means those particular code aspects have been removed. Something with a sign of plus, it means a new program is added, and so on. So to actually do that, you need to have a registered email ID. That's all you need for getting the GitHub. So where you can add your name and a particular email ID. If you say, we usually say it as a config because you're configuring GitHub in your system, saying that this is the particular username and this is the email ID, so that whenever you want to upload or update, it will understand that these programs or contents belongs to this particular user. It is just like a simple authentication. Having said that, so now you have to say you have a folder where you have 3 or 4 programs, and how to actually put it into a GitHub repository. So what you actually do is, you have something, it's called a git bash, which is also provided by the GitHub during your installation. So you will go to this command and you will say git init, which means you're initializing a particular project or your a particular repository that you want to actually save and keep track of. And once you do that, once you initialize it, you can then add new files, saying either saying git add that particular file name, or git add space dot, which means it will add all the files in your current project. And once you have added all your current project, you will do a git commit. Git commit is nothing but a particular description or a message that is useful for you as a landmark, or as a placeholder, or as like a bookmark that you use, where you say what is the purpose of it, or what are the changes you have made, in case if you're updating your particular repository. So if you see, there are 3 key aspects. First is initializing, second is addition of the files, and third is commit. You initialize only once for a particular project. Later on, depending on the project's scale, you will be adding or removing files based on your necessary. But commit is something you do regularly after making those changes, so that you understand what particular changes are made, what is its importance, and so on. And then, so this is, for example, is a functional structure where you have a working directory where you have your 4 or 5 programs that you are using to analyze certain data. So when you do a git addition, it is called a staging place. It means that you have added these files, but they have not pushed into your GitHub repository. So by when it is in staging section or staging area, you can still change or make modifications to it. But once if you have committed, you can't make modifications for that particular time. So if you want to again make modifications, then you have to again do a after making changes, you have to again go back and do a git add and then do a git commit. So once you have made a git commit with a particular message, that message will stay on for the changes that you have actually committed for that particular purpose or an objective. So the next thing is git pull or git clone. There would be many as the earlier speakers have also mentioned. It's a large community, so many people would have developed a lot of programs which are quite helpful for us to do certain analysis. You can either do a git pull or a git clone from their repositories if they are in public, so that once you have done that, you can download their entire project structure and you can use it. So if you want to push your or upload your project directory, then you use the command called git push. So git push is for uploading and git pull is for downloading. Git clone means if you want to take any person's project directory and or anybody's programs, if you want to use, you can do a git clone citing their user name. So this is another additional command called git remote add, which means that you are actually adding your particular project name, so that each project directory will have a single origin link that you would be presenting. It is like you can consider it's like a car key or a bike key. So for every vehicle you have a unique key. So similar to that, you will have a particular link for every of your project repository that has been uploaded. So just to show you as a very a fair demo of it. So the messages that you are actually seeing here is the messages that have been added by the particular user saying what the change he has been made for that particular program that he has uploaded uh, in his project directory. So if you see, there is a green color icon that is being listed here. If you click that, you will get a particular section to it. You can either copy this link, paste it in your GitHub, Git Bash, which is called as a terminal, and you can download their entire project repository. Else, you can just click the download zip icon where it will zip the entire project content and it will download it to yours. So if you see here one more thing, so this is the particular user place, and you can see here the list of projects that he has done. So and then you see public means it is available for people to download and use. There are certain aspects called private also, where you don't want to share it with the public, but it's for your personal condition. So, and you can also track what changes you have made every month, how many changes you have made, how is your contribution in the public space that is going on. So GitHub repo is irrespective of language. You can download, you can save, upload, keep track of your programs irrespective of language that you are using. It can be either C, it can be either Python, it can be either R, any programming, that doesn't matter. Similarly, you can use a number of files. You can use a JavaScript, you can use a notebook pad, you can use a standalone script. So any of the scripts you can actually keep on updating. So there's something called projects. So projects, if there is anything in public, it would be listed, or you will have something called repositories. The repositories is also a form of a mini project, but it would be specific to certain application that the user wants to solve. So you can actually have a simple description, a simple readme for it, and you can actually explain it so that the person who actually wants to use it can follow the instructions and do, or can understand what the project directory is been doing. So one of the biggest advantages that the GitHub is being useful. Say you are working as a research scientist, you are working or you are working in either in a company. You will have certain releases for publishing certain articles. You will have certain releases that you have been pushing out to public rollouts. So there you can use the version numbers, and then you will know based on the version numbers what things have been made. So for example, you can see here with the information that is being displayed, the user has totally made 209 commits. Okay, and this is the commit ID. So you can, if you want a particular commit, or a previous version, you can just give the commit ID and you will get the you can get the project structure that was being uploaded before. So this helps you to develop your own project stage wise, and you can also maintain the version for every stage wise. Having said that, the GitHub is not only limited to that. I will show you one of the GitHub profiles which have been used as a project portfolio as well. So say this user has n number of projects, but how to demonstrate it in a useful way so that when a person from an industry or from an acad who is reviewing your application, or who wants to connect with you, will know what work have you have done. So GitHub will allow you to do even those things. Okay, so this is one of the GitHub pages of a person. Okay, so he has mentioned what all projects he has done, and he has also mentioned what is his education, what is his experience. It is like a simple resume build, but he has done it using GitHub. So when a person is looking at your GitHub profile, like your LinkedIn profile, he will know what kind of projects you have done, and what are the and the biggest pluses. He will actually know what exact programs you have written, how is your programming knowledge, whether the things that you have mentioned in your resume is actually being reflected in your profile or not. So that gives a more better overview for a person who wants to connect with you, either for a job or for an internship, or for an acad purpose. So this gives a more it's like it will give you a more, it's like a portfolio that you have, that you want to share it with the external community. So this is all about GitHub. Obviously there are a lot of commands and techniques within the GitHub, and it's a continuous learning process. It is not like a short course, but once you actually start and using with it, there are a lot of commands, but these are the basic commands that I have put forward, that will actually enable you to start and operating your own GitHub profile. So, having said that, the next most important platform that has probably revolutionized the field of data science and machine learning from 2010 is Kaggle. Kaggle is actually developed by Google, but it is I'm telling you, it has just changed the face, the speed, and also helped lots of people, lots of developers, a lot of researchers who want to do progress in their research in their own fields. So Kaggle, so this is how a Kaggle actually website looks like. So similar to GitHub, you should also register for Kaggle, but it's more of a simple registration. You can do it with your email or your Google account. So Kaggle hosts a wide range of things. One is it hosts a lot of open source data sets. So and if you want something, some cancer data set, okay, so it will give you the list of whatever data sets that are associated with cancer. So depending on what kind of a data you want to work on with, you can use this particular data to do your own research. So and all these data sets are for non-commercial purposes. You can use them for free. It gives you the resources to use this data to run the programs in the same platform, so that you can actually see what can you extract from the data, what can you learn from the data. Not only that, you can also see a lot of models that have been developed using these data sets. Okay, uh, say for example, in cancer itself, you want to see certain things. So there are already models that have been developed by on breast cancer, on cancer cells, on skin cancer, and so on. So you can actually go to those particular models, you can see what they have done, and you will also have saying that these models have so and so accuracy, and what the model does, how to use it, what are the characteristics of that particular model, like how the model is being built, what kind of architecture it is being used, and on what particular data set it is being used. So this gives you a lot of uh, uh, a good starting point. Say you need not build it from the scratch, but you can take from a good take certain things from a very good position, and you can build things over the top, so that your time from building from scratch to your time from using something resourceful and reaching your goal would be reduced, and it will also give you an additional perspective on how the other things how other people are actually coding, what kind of logic they are using, what kind of models they are using, what they have achieved with the models, okay, and what is the drawback of that particular model, and what you have to think different. So all these kind of a mini literature survey you can do in the Kaggle. So this gives you a great head start when you're compared to start doing from the scratch. And also said that you can also push your notebooks here. You can give your you can actually connect your Kaggle profile to your GitHub, so that the moment you update in one place, it will be maintained. You can use these places, create the notebooks. They also provide a lot of computational resources, because when you are working with a big data, right, our standard laptops or certain types of systems that we are using may not be able to handle the data or the process effectively. So you need larger computational systems with larger power to execute your functions or your programs, or to do training of your certain models. So for that, the Kaggle gives you the access to its resources, where you can run your notebooks on those resources, and you can get the results. So you can also do that through Kaggle. And most importantly, it gives you a lot of courses. Okay, certain courses for people who are very new to programming, new to machine learning, new to feature engineering. Certain people want to do on databases. So all these kind of certain courses, certain are free but certain are paid. But it will try to give you certain basic overviews about how things have been done, certain basic concepts that you really have to learn. So these kind of things you can use, and it's extremely helpful through this platform. And the most important thing, having said all of these things, the most interesting aspect is the competitions. So it also hosts wide variety of competitions, where you can submit your code, collaborate with people, and participate in in a hackathon, try to win prizes. This gives you three things. One is the opportunity to see how others are performing. It will give you a larger scope to learn faster. And obviously, a competition, a good healthy competition is always encouraging for the personal and the knowledge growth. So this plays a very very key role to it, and their prices are also quite famous and hefty for you to earn. So the Kaggle, this, having said about the Kaggle, this is like a very important landmark in the data science and the machine learning thing. So it helps you to update your skills, use public notebooks, and especially collaborations. You can collaborate with a lot of people who are working on a similar goal. And also, the best thing is to get the data sets. Certain times certain data sets are very private. You need to get permissions to use it. But you can this gives you a room to use a lot of public data sets, which have been properly been acquired with the proper experimental paradigms. So that is something great to use with. So having said that, the another most important one is the LinkedIn learning. So we all build our profiles in LinkedIn, but having said that, GitHub and Kaggle profiles are also, even though they are good, LinkedIn is something supreme of it, because it consumes all of these data, and it projects you in a space, in a dimension that will boost your career aspects and your learning capabilities. The LinkedIn also comes up with certain programs and courses that helps you to learn basic coding, basic presentation skills, certain management skills that are required. And you can also get a certifications from LinkedIn, saying that you've been certified in machine learning, you have been certified in using Microsoft Word, you're certified in Linux, you're certified in coding, and so on. And you have a quiz, you can register for the courses. Obviously, some of the courses are paid courses, so you have to choose wisely depending on what you want. One good thing with LinkedIn is it gives you a more personalized courses depending on your profile. That would be helpful to boost your career and future opportunities. That which is something that lacks in Kaggle and so on. That is something great with LinkedIn. So as I've told you, personalized recommendations, you can have your own schedule. and all these courses are being taken by industry experts. So that is something great. And The most important thing is you can save these certain videos. You can go back, see at your own preferred timings, and you also have certain transcripts that have been generated with these videos, which will help you to give a good audio as well as a textual information that you can use it for the later purposes. Having said that, you will have a quiz at the end of the course, and that will give you an essential certificate, which can be used to be posted in your LinkedIn profile. And Some of these certificates also hold a good value, if you're looking for certain internships or job opportunities. So, having said that, even Biotechnica offers a lot of courses. I would recommend a lot of you you who are new to this platform to follow some of their content on YouTube and their courses. They offer a lot of courses for biology in terms of data science and AI, and which are very useful and recommended as well. So please do have a look at them. So having said that, there is a new fear or a rumor that's saying that the AI will take away all the jobs and so on. I strongly condemn that. AI is to help us, is to assist us. So as I've told you, the start of the presentation also, that there are certain things that are very repetitive in our physics lab or a chemistry lab. When we try to take data, take notes, certain things are very repetitive. You have to do it for every experiment and something. But with the help of computers, you need not need not manually open certain files and do certain tasks. You can make certain things automated. Say for example, you want to analyze a probability or do certain p test or a t test. So you have certain data, you want to access certain column, you want to focus on only on specific kind of a data. So then you can use this pandas library. You can read your CSV files where you can choose specific column on which you want to do analysis on technique, on which you want to apply certain statistical methods, so on. So those kind of things, the automation helps you a lot. The best thing that automation does you is it will get you a good starting point, so that you can start applying our mind to analyze the data and get the key insights out of it. If not, if you're doing it manually, a lot of time will get consumed. The time where you start inferring the knowledge or the data, it gets too late. And we are running in a fast paced environment, so every minute matters. So automation is something that will help you to save help you save time, give you a good head start in getting a good inference and understanding the analysis that you have done. So that is why automation is different. Domain expertise is different. Both are not the same and cannot be compared. So that's why AI is more in the automation side, so that it will give you good assistance in terms of analyzing your data. And once you get that analysis done quite quickly, you can use your domain expertise to actually infer the knowledge from the data analysis or visualization, both on that. And then you can give out your insights based on the experiments that you have done or you have conducted over a period of time. So, having said that, this is something a continuous process. This is not like a short course, as I have repeated before. You need to keep practicing, keep upgrading yourself. Once you stop upgrading or start being stagnant, you cannot progress. It's not only in biology, but it's applicable to all fields. So you have to keep updating yourself, keep projecting yourself in a very good way. Do remember these kind of profiles, so you can actually put out saying that you have interned, you want to work, you're projecting different projects what you have done. This will open a lot of collaborations over a period of time, and will shape your career in a very very different way. Also try to look at certain research articles that have been put out in the field, especially to areas where you are more interested on. Be updated and do it. And moreover, focus on a proper story telling, what you want to infer, or what are your insights from these data or your research. That is more important, so that you can only get once you save a lot of time in using the automated tools, and also to rigorous practice and so on. So anyway, I want to end my talk here. I would like to thank everybody, the previous speakers, and the wonderful audience, for being and spending your time here. So thank you once again.
Thank you so much, Mr. Kapardi, for that incredible session. I'm sure students have found it very valuable. And I also take this opportunity to thank all our brilliant speakers for the insightful presentations, for such insightful presentations. So, what we learned today, let me give you a quick recap what we have just explored. Dr. Nilofer Sheikh laid down a strong foundation by showing us in detail how AI/ML tools like Python, Jupyter Notebooks, AlphaFold, and more are revolutionizing the way we analyze biological data, especially in genomic and transcriptomics. Dr. Ishan Pandey took us further into real world biomedical applications with tools like Cell Profiler and KNIME, showing us how AI is transforming precision medicine and image based analysis. Lastly, Mr. Kapardi wrapped it up all beautifully by focusing on career success, how to leverage platforms like GitHub, Kaggle, and LinkedIn Learning, and also how to build an AI driven resume that truly stands out. His take on balancing automation with domain expertise was especially timely. So, we have we have exceeded our time, unfortunately. Yes, so we'll be taking only three of the questions, which was already asked by from some of our audiences. Okay, so one question we'll take from the new question, recent question from Pavni Annam Botla. Good evening, sir. Please suggest some certifications related to this domain. Mr. Kapardi, do you have any take on this? Any certifications related to this domain?
You can start looking at Kaggle's certain certifications, uh, and you have certain, as I told you, even Biotechnica gives certain courses. You can have a look at their website. They have more detailed, things. Probably you can choose and you can interact with them. So I would suggest these two. More on the certification, also you can focus on your project portfolio building using open data sets, what you're doing. That is also a good head start.
Okay, thank you so much. First of all, I would like to inform our audience that we have already exceeded our time. So, we'll be taking maximum three more questions. So, I request our students not to send any more questions now. And if you have any questions, you can always mail to cst@biotechnica.org. It will be given to you now in the chat box. cst@biotechnica.org. You can message your queries, and we will be getting back with the reply from our experts. Okay, so the next question is from Irfan A. Khan. He says, I'm from PCB background. What should I choose? BTech Biotechnology or Bioinformatics? So I would request Dr. Ishan Pandey to throw some light on this question. I am from
Yeah,
PCB background. What should I
yeah.
choose? BTech Biotechnology or Bioinformatics?
No, I think BTech Biotechnology would be a good option rather than bioinformatics. If you do study BTech Biotechnology, that will give you a basic core analysis of biotechnology and the use of bioinformatics in biotechnology. So bioinformatics will be an integral part of BTech because it is a course on technology, with the bachelor's of technology. So bioinformatics will be your core, but in your masters you can go for bioinformatics, or in your PhD you can. It will be more better if you do a research or bioinformatic analysis, because nowadays it is about interdisciplinary research and BTech bioinformatics. When you do BTech bioinformatics or you do a bachelor's in bioinformatics, then it will be very hard to proceed in jobs. But having a BTech in biotechnology can give you some more edge, having some job aspects as well as your research career in bioinformatics as well. I hope I'm clear.
That was helpful. Now, next question is from Saurav Bakshi. He says, Ma'am, any summer internship programs for applying? I am pursuing second year BTech Biotechnology. So, Nilofer, ma'am, would you please like to answer this question? any summer internship programs?
Yes, ma'am. Uh, so it is totally depend on what kind of skills you want to upgrade accordingly. So if you are in related experimental stuff, so in that they are providing you an hands-on experience to get to know how which kind of tools and techniques you can process with the wet lab considerations.
Okay, ma'am. Thank you for that answer. That's the last question now from Bandana. Ma'am, can you please tell me a little bit more about BioPython in managing biological data? So this would be the last question for the day. Ma'am, please throw some light on this question. Nilofer, ma'am.
So basically, in the context of BioPython, as I mentioned previously in the slides, that it is specially designed to work with respect to your biological data sets. So if you want to process with respect to the programming stuff and you want to integrate the biological data in the form of the pipeline, then BioPython will aid you in that. So BioPython will aid you in processing the data like performing a simple sequence analysis such as blast algorithm. I hope you know about that, right? So blast is one of the web-based tool, but using BioPython we can do that stuff with programmatically. So you can perform more specific analysis, you can even move ahead and you can perform the genome analysis. So we have certain packages for the same. You can even come up with a data visualization so that you can find a personalized data sets and even the type of data visualization what you're expecting. For example, if you're using blast database and certain kind of visualizations are not available, so you can regenerate those things personally with respect to your data stuff. And then you can even proceed with respect to the 3D structure visualizations for publication quality images, and many more things are available. I think that is a really good to proceed with BioPython and explore how you can deal with your biological data and their applications, and even look for the publication quality work.
Okay, ma'am. Thank you for the answer. I request all the audience, if you have any more questions, the email ID is on your screen. Kindly note it down, cst@biotechnica.org. We would love to answer your questions. Do write to us and we will get the replies from our experts and we will send back to you. With that, I would like to extend a heartfelt thank you to all our brilliant speakers for the day, Dr. Nilofer Sheikh, Dr. Ishan Pandey, and Mr. Kapardi. It was lovely to have you here, and our students benefited a lot with your sessions. Thank you to all the attendees for your active participation and great energy. Stay curious, stay connected, and we hope to see you in our next session soon. Thank you, everyone. Good night.
Thank you, everyone. Thank you.
Thank you. Thank you.